You know, usually when we talk about treating a disease today, there's this expectation of absolute precision.
Right, like a targeted strike.
Exactly. Like if you have a bacterial infection, you take a specific antibiotic and boom, it's handled. But then you look at something wildly complex like Crohn's disease.
Yeah, which is a whole different ballgame.
It really is. I mean, we have these incredibly advanced biologic treatments now, right?
Yeah.
Drugs that hunt down the immune system's specific chemical messengers.
The cytokines.
Right, the cytokines. Yet, despite these modern medical marvels, a staggering 15% of Crohn's patients still require surgery, like surgical removal of parts of their intestines within 5 years of diagnosis.
Which is just, it's a profoundly frustrating clinical reality.
Yeah.
We have these blockbuster drugs that neutralize inflammation, but for a huge chunk of patients, the gut just continues to destroy itself.
It's like we're missing something massive.
Exactly. It suggests our understanding of the disease at a foundational level is just, well, missing some crucial pieces of the puzzle. We're treating the symptoms, maybe even the mechanisms, but completely missing the root environment.
Which brings us to our mission for today's deep dive. We are looking at a groundbreaking new study published in Nature Genetics that actually attempts to map that exact root environment.
And they call it the IBDverse.
The IBDverse, yeah. It's this unprecedented single-cell RNA sequencing atlas of the terminal ileum.
Right, so that's the very end of the small intestine.
Exactly.
Yeah.
Which is the exact spot where Crohn's usually strikes the hardest. So for you listening at home, whether you're a medical professional or maybe navigating IBD yourself or just, a massive biology nerd, we are going to unpack exactly what goes wrong in the human gut at a microscopic level.
And the scale of this atlas is just, it's what makes it so revolutionary. I mean, they didn't just look at a handful of samples.
No, it's huge.
Yeah, they sequenced over 1.1 million individual cells.
Mind-blowing.
From 343 individuals. So that's 111 Crohn's disease patients compared directly against 232 healthy controls.
Okay, let's unpack this. Because to really grasp how they mapped 1.1 million separate cells, we have to talk about the technology itself, right? Single-cell RNA sequencing or cRNA-seq.
Right.
So think about traditional tissue sequencing, like making a massive fruit smoothie.
I love this analogy.
Right. So you take a sip and you try to guess the exact ratio of strawberries to blueberries to bananas. You get the average taste, the dominant flavors, but the individual ingredients are totally lost in the blend.
Yeah, they get mashed together.
Exactly. But single-cell RNA sequencing is entirely different. It's like looking at a beautifully arranged fruit bowl. You can literally pick up every single blueberry, every strawberry, and examine it individually.
And if we take that fruit bowl analogy a step further, in a smoothie, the loud dominant flavors completely mask the rare one.
Oh, right. Like if you put too much banana in?
Exactly. In bulk sequencing, the loud signals from common structural cells just drown out the whispers of very rare, potentially disease-causing immune cells.
Makes sense.
So by looking at the individual components of the bowl, we can sequence the RNA transcripts of single cells. And remember, DNA is the static blueprint of the cell, but RNA
tells us what's actually happening.
Right. It tells us which parts of that blueprint are actively being read and built at that exact second. We see exactly which genes are turned on or off.
The logistics of that sound like a nightmare, though. I mean, if you try to pull a single cell out of a piece of living human tissue, you're going to stress it out, right?
Absolutely.
And if a cell gets stressed, it turns on stress genes. So suddenly your RNA data is just the map of a cell panicking, not an actual map of Crohn's disease.
That is the exact hurdle. Getting the cells out without altering their genetic behavior is notoriously difficult.
So how did they do it?
The methodology here was incredibly meticulous. During colonoscopies, they took what are called pinch biopsies of the gut. And the immediate step was to plunge that tissue into a chilled solution on ice.
Ah, so that effectively freezes the cell's biological clocks.
Exactly. They hit the pause button before the cells even realized they were removed.
That's brilliant.
Then they introduced a highly specific cold-active enzyme called Bacillus licheniformis protease.
Wow, say that 5 times fast.
Right. But because it works at low temperatures, it gently coaxes the cells apart without waking them up to trigger that transcriptional panic we talked about.
Oh, wow.
Yeah, and they follow that up with collagenase to dissolve the structural matrix, and suddenly they had a pure suspension of individual, totally relaxed cells.
Which allowed them to map, 57 distinct cell clusters?
Yes, 57 highly distinct cell clusters across the gut's lining, the immune system, and the structural tissue.
Okay, but wait. I was reading through the supplementary data on those clusters, and I noticed a detail that really made me pause.
So the replication rate.
Yes. So the researchers were incredibly rigorous, right? They split their 343 individuals into a discovery cohort and a replication cohort to basically double-check their own homework.
Which is good science.
Definitely. But they found that only 44% of the differentially expressed genes, like the genes they flagged as acting abnormally in Crohn's, actually replicated in the second group of patients.
Right.
When I saw that, I thought, if this tech is so advanced, why is the replication rate less than half? Is the technology flawed?
Well, what's fascinating here is that it's a totally valid concern, but it actually points to a broader phenomenon known as the reproducibility crisis in single-cell genomics.
Okay, so it's a known issue.
Yeah, and this paper is quite brave for confronting it head-on. The 44% replication rate isn't a failure of the tech. It's actually a profound revelation about a concept called transcriptional plasticity.
Transcriptional plasticity. Okay, what does that mean in plain English?
It basically means immune cells aren't static robots. They are incredibly reactive. They change their genetic behavior based on a vast array of subtle environmental cues.
Like what?
Like what you ate for breakfast, your baseline stress levels, a poor night of sleep, or even minor differences in medication.
So the cells are just constantly shape-shifting.
Yes,
Which
exactly.
makes pinning down a universal, like, Crohn's signature incredibly difficult.
It does. And beyond the plasticity, some of the critical immune cells they identified are just astonishingly rare.
How rare are we talking?
For instance, they flag specific subsets of inflammatory monocytes and macrophages, cells carrying markers like S100A89 or CXCL910.
Okay.
In a healthy gut, these specific cells make up less than 2% of the myeloid immune compartment.
Wait, less than 2%? They're practically ghosts.
Exactly. So when you're dealing with populations that small, trying to compare them statistically across hundreds of different human beings with different life experiences and diets is mathematically daunting.
Yeah, I can imagine.
So replicating 44% of those disease signatures across two massive cohorts is actually a triumph of their rigorous design.
Okay, so let's do a quick recap of what we've covered so far. We've got this massive IBDverse atlas using single-cell RNA sequencing to look at 1.1 million individual cells. They had to use a super delicate cold enzyme process using Bacillus leishmaniiformis protease just to get the cells out without stressing them.
Spot on.
And we learned that immune cells have massive transcriptional plasticity, shifting constantly based on environment, making it hard to find consistent signatures, especially in super rare cells making up less than 2% of the tissue.
That's the perfect summary.
So here's a question for you listening right now. Think about your own body's environment. How much do you think your daily stress or diet is shifting the plasticity of your immune cells right this very second? It's kind of a wild level of microscopic chaos.
It really is a dynamic ecosystem. And understanding that chaos is vital as we move from the technology itself into the actual physical structure of the gut, which is where this disease really takes root.
Okay, so let's dive into that physical structure, the gut lining or the epithelial compartment. The scientists call this area the crypt-villus axis.
Right, which sounds complicated, but it's really not.
Yeah, to visualize this, I want you to imagine a factory conveyor belt. Down at the bottom, in these little valleys called crypts, you have your stem cells.
The factory workers.
Exactly, the factory workers constantly producing new lining cells. As these new cells are created, they get pushed up the conveyor belt onto the villus. These are the finger-like projections that absorb nutrients from your food.
Yeah, reaching out into the gut.
Right. And eventually, the cells reach the top, they die, and they shed off into the digestive tract. It's this constant cycle of renewal.
In an active Crohn's disease, that entire conveyor belt is essentially on fire.
Wow.
Yeah, the researchers observed a massive upregulation of interferon alpha, beta, and gamma signaling all along this axis.
And interferons are like the chemical alarm bells of the immune system, right?
Yeah, exactly. When they ring, they trigger cells to drastically upregulate a protein complex called MHC-I, or major histocompatibility complex, class I.
Okay, let me offer a visual for MHC-I because it is such a core concept for you to understand. Think of MHC-I as a cell holding up a wanted poster to the immune system.
I like that.
The cell basically takes little pieces of proteins from its own interior, mounts them on this MHC-I poster board, and shoves it through its outer membrane.
Right, for everyone to see.
Yeah. So roving immune cells, specifically CD8 plus T cells, patrol the tissue and inspect these posters. If the poster shows a healthy, normal protein, the T cell just nods and moves on.
Nothing to see here.
Exactly. But if the poster shows a viral protein or an abnormal bacterial fragment, the T cell immediately kills the infected cell to protect the body.
That captures the mechanism perfectly. Now, during an active Crohn's flare-up, every cell on that epithelial conveyor belt is holding millions of these wanted posters, screaming for the immune system's attention.
So the tissue is inflamed, the immune cells are attacking, the gut is taking heavy damage.
Yes. But the most critical finding in this entire paper is what happens when that inflammation finally stops.
Here's where it gets really interesting.
Yeah, so when a patient is treated and goes into clinical remission, a doctor will often look at their gut through an endoscope. To the human eye, the tissue looks perfectly pink, healed, and healthy.
Right, the macroscopic inflammation has totally resolved.
Exactly. However, when the researchers looked at the single-cell RNA level of these seemingly uninflamed, healed tissues from Crohn's patients, they found that this MHC-I alarm system stayed permanently elevated. Permanently. And it was elevated specifically in the MKI-67 plus stem cells down in the crypt.
So wait, the factory workers at the bottom of the conveyor belt?
Yes.
So even though the fire's put out, the factory workers are still holding up the wanted posters?
Yes, they retain what we call a molecular scar. The macroscopic tissue heals, but the stem cells hold an epigenetic memory of the trauma.
That's insane.
And because those stem cells are the ones constantly producing the new lining of the gut, it means the entire newly built gut lining is born permanently primed for a fight.
They're basically born paranoid.
Paranoia is a very good way to describe it. Because they have so much MHC-I on their surface, these cells might be engaging in cross-presentation of luminal antigens.
Meaning what exactly?
Essentially, they could be taking innocent particles from the gut contents, like regular food or normal microbiome bacteria, and presenting them to T cells as if they were dangerous pathogens.
Oh, wow. Which completely explains the chronic nature of Crohn's disease. I mean, we always talk about it as a chronically relapsing condition.
Right.
If the stem cells themselves are scarred with this memory, the gut is never truly at peace. It's a traumatized environment, just waiting for the slightest excuse, a minor infection, a period of high stress to trigger a full-blown immune attack again.
It really forces us to entirely reevaluate the clinical concept of remission.
Yeah.
Is the patient actually healed, or is the tissue just temporarily quiet while maintaining this baseline level of hypervigilance?
That is a really tough reality to swallow for anyone managing an autoimmune condition. It blurs the line between being better and just being, between flares.
It does.
So let's quickly recap part 2. We talked about the crypt-villus axis, our cellular conveyor belt. Interferons trigger MHC-I wanted posters. But the huge takeaway is that even in remission, MKI-67 plus stem cells keep those wanted posters up, carrying a permanent molecular scar that causes them to overreact to normal stuff.
Exactly.
So here's a question for you listening. If our stem cells have a quote-unquote memory of past trauma, how does that change the way we think about the concept of remission?
It's a profound shift in perspective.
It really is. But if the epithelial cells are the ones holding up these wanted posters, they have to be showing them to someone, right? The tissue doesn't just spontaneously combust. The immune system has to pull the trigger.
Which brings us to the immune compartment, and specifically to a type of cell called a macrophage.
The macrophages, these are the Pac-Men, right? The security guards of the body. They roam around the tissue gobbling up debris and pathogens.
Normally, yes. But the data shows that in Crohn's disease, a very specific, transcriptionally distinct subset of these security guards goes completely rogue.
Rogue security guards.
Yeah, they're marked by a gene called ITGA4. In a healthy gut, these ITGA4 plus macrophages make up about 10% of the myeloid immune cells. But in an inflamed Crohn's gut, they expand to 16%.
Wow. Okay.
And their danger isn't just in their numbers, it's in their behavior. The single-cell sequencing revealed that these ITGA4 plus macrophages are acting like massive factories for pro-inflammatory cytokines.
The chemical messengers again?
Right. They are pumping out IL-6, IL-12, IL-23 alongside complex inflammatory loops involving IL-10 and IL-20.
Okay, we mentioned cytokines at the top of the deep dive, but let's break down how those specific chemicals actually cause the damage, because the researchers mapped out the internal wiring these cells use to communicate, didn't they?
They did. If cytokines are chemical text messages sent between cells, the IL-6 or IL-23 text message hits a receptor on the outside of a neighboring cell.
Okay, cell gets a text.
Yep. This activates an internal communication relay known as the JAK-STAT pathway.
JAK-STAT, okay.
You can think of JAK, specifically the JK2 protein in this context, as the cellular antenna that catches the signal.
Got it.
Once JK2 is activated, it phosphorylates, which basically means it wakes up, a set of messenger proteins called STATs, specifically STAT 1, 2, and 3.
And what do the STATs do?
These STAT proteins then dive directly into the cell's nucleus, bind to the DNA, and alter its expression to turn up the volume on inflammation.
Okay, the reason this is so crucial is that this is where the microscopic biology meets the actual pharmacy counter, right?
Yes, exactly.
If you or someone you know has Crohn's, this pathway is exactly why certain medications are prescribed. For example, vedolizumab is a widely used biologic drug.
Right.
It is literally an antibody engineered to bind to and block IgA4. And it also explains the recent rise of JACO inhibitors, which are a newer class of oral medications designed to just jam those cellular antennas we just talked about.
Exactly. We are not just looking at abstract biology here. This atlas is mapping the exact cellular targets of these blockbuster drugs in high definition.
That is just so cool to see mapped out.
And the atlas also reveals how these rogue macrophages coordinate their attack. The data shows that these ITGA4 plus macrophages drastically upregulate genes for structures called immunoproteasomes, specifically genes like PSMB8 and PSMB9.
Wait, let me guess how that works, because I know a regular proteasome is essentially a cellular garbage disposal, right? It breaks down old, misfolded proteins so the cell can recycle the parts.
Right.
But the immunoprefix implies this one is doing something specific for the immune system. Is it processing the proteins for the wanted posters?
That is spot on.
Oh, wow.
Yeah, if a regular proteasome is a garbage disposal, an immunoproteasome is a high-security paper shredder.
Okay, I can picture that.
It specifically cuts up invading viral or bacterial proteins into the exact right molecular size so they can be glued onto the MHC-I wanted posters we discussed earlier.
That is sinister.
Right. These ITGA4 plus macrophages aren't just shouting at the tissue with cytokines. They are actively manufacturing the physical evidence needed to keep the adaptive immune system in a state of absolute fury.
Okay, so let's recap section 3. We've got rogue security guards, the ITGA4 plus macrophages, expanding their numbers. They're pumping out inflammatory texts like IL-23, jamming the JAK-STAT antennas using JK2 and STAT proteins, and they're running the PSMB89 paper shredders to frame the tissue as infected.
Perfectly stated.
And we know this is real because drugs like vedolizumab and JAK inhibitors specifically target these exact mechanisms.
Exactly.
So, question for the listener. Knowing that specific cells like ITGA4 plus macrophages are driving the inflammation, do you think future therapies should focus on neutralizing the cytokines they release, or should we be trying to eliminate these specific rogue cells entirely?
It's the multi-billion-dollar question in pharma right now.
It really is. It paints a terrifying picture of the disease. But it brings up a classic chicken or egg scenario. How do we know if these rogue macrophages and scarred stem cells are the cause of Crohn's disease or just a symptom?
Right.
Like, are they the arsonists who started the fire, or are they just the firefighters caught in a burning building reacting to the heat?
Establishing causality is always the hardest part of observational biology. To tackle it, the researchers utilized GWAs.
Genome-wide association studies.
Exactly. Over the past couple of decades, scientists have scanned the DNA of hundreds of thousands of people and identified specific genetic typos or variants that statistically increase a person's risk of developing Crohn's.
So these are inherited risks you're born with.
Right. What the researchers did was take these known population-level genetic blueprints and map them directly onto their single-cell IBDverse atlas.
So they wanted to see exactly which cells in the gut were actually reading the faulty blueprints.
Precisely. And the mapping was incredibly specific. For example, they looked at a gene called FUT2. Mutations in FUT2 are heavily linked to a disrupted gut microbiome.
Okay, where did that map?
The atlas showed that this gene is expressed almost exclusively in the enterocytes and stem cells of the epithelial lining.
Well, that makes sense since the gut lining interacts directly with the microbiome. But what about the heavy hitters? Like in Western populations, the strongest known genetic risk factor for Crohn's is a mutation in the NOD2 gene. Where did that blueprint map to?
This is where the precision of single-cell sequencing truly shines. NOD2 didn't just generally map to the immune system. It mapped specifically to those very rare monocytes we mentioned at the beginning, the ones making up less than 2% of the compartment, like the S100, A89, and SOD2 positive ones, and to immature macrophages.
Wow.
Importantly, the NOD2 risk gene was not expressed by the resident, peaceful macrophages that normally live in the healthy gut tissue.
Wait, really?
Yes. The genetic defect is carried specifically by the circulating immune cells that are recruited to the gut from the bloodstream during an emergency.
So the troops being called in as reinforcements are the ones carrying the faulty programming.
The data pushes that even further. They performed a statistical test looking for heritability enrichment.
Which means what?
Essentially, they asked, if we bundle all the genetic risk factors for Crohn's together, which cell types carry the highest concentration of this inherited risk?
Okay.
The heritability enrichment was only significant in the immune cell types, the monocytes, the dendritic cells, the specific macrophages. The inherited risk was not significantly enriched in the epithelial cells.
Wow. Okay, let's recap this and synthesize the whole picture because this is huge.
Go for it.
So GWS genetic blueprints were mapped to the cRNA-seq atlas. FUT2 went to the epithelial cells. But NOD2, the biggest risk factor, mapped only to rare circulating monocytes and immature macrophages, not the resident ones. And overall heritability enrichment was only in the immune cells. So what does this all mean? The epithelial cells, the gut lining, they carry the molecular scar. They hold the epigenetic memory of the inflammation and remain permanently paranoid. But the immune cells carry the genetic loaded gun.
That's a great way to put it.
You might have a perfectly normal, genetically healthy gut lining, but if your circulating immune cells are genetically hardwired by NOD2 and ITGA4 mutations to overreact, they will assault the tissue. And that assault causes the damage that leaves the permanent scar on the stem cells.
Exactly. The epithelial barrier is the victim that learns to be hypervigilant, but the immune system is the genetically predisposed instigator.
Man, that is just incredible to see it laid out like that.
And knowledge is most valuable when it's applied. What this 1.1 million cell atlas does is fundamentally shift how we approach the disease.
Yeah.
We can no longer treat Crohn's as a monolith. We can now see the exact molecular dialogue between a genetically risky macrophage and a traumatized stem cell. It provides the foundation for creating highly targeted, personalized therapies where we aren't just suppressing the whole immune system, but aiming at the exact cell at the exact right time.
And it really highlights how combining different perspectives, right, bringing together population genetics, single-cell RNA sequencing, and clinical endoscopy, they just enrich our understanding so much more than any one discipline could on its own.
It's a masterclass in modern systems biology, truly.
It really is. So to wrap up our deep dive today, I want to leave you, the listener, with a thought to mull over. We started by discussing how standard treatments still fail 15% of the time, often leading to surgery.
Right.
We now know that even when those treatments seemingly work and the visible fire is put out, the gut's stem cells hold on to a permanent molecular scar, constantly holding up those MHC-I wanted posters.
The paranoia.
Yeah, the paranoia. So if inflammation leaves this permanent cellular memory, could the true future of curing Crohn's disease lie not just in blocking the immune system's weapons, but in discovering a way to medically erase that cellular memory? Like, can we find a way to convince the gut stem cells that the war is truly over?
That is the next great frontier.
It's fascinating stuff. We'll keep questioning, keep exploring, and we will catch you on the next deep dive.